Gosh, it's so dark in here, both shut. And
Yeah.
I shut them trying to get more heat. I've got 2 space heaters going too, so hopefully.
Thank you.
I mean, it is 5 degrees warmer than what it was when I came in here first thing this morning. So anyway, let's, I'll back up a couple slides just because I want to look at structure for just a minute. I know this is a class where we're learning more about things, but I want to look at the relationship of the phosphorus with the double bonded sulfur. And so in that, if you'll notice, that piece of the molecule is the same, that piece of the molecule is the same, and so that's the working part. And you notice that we can change a part of the molecule structurally to still get the same effect, acetylcholinesterase inhibitor. But if we go on and change that a little bit more, if we start looking here, then what we can find, here's that phosphorus, here's that oxygen, right? And the sulfur here, we've changed this is still that phosphorus-sulfur interaction, but we've changed this part of the molecule, and it's made it much less toxic for humans. Okay, you guys with me? And so, in that relationship, then that molecule is much safer. If we start looking at the carbamates, so carbamates are also acetylcholinesterase inhibitors, but they don't have the phosphorus in them. Okay, you guys with me on that? So the structure changes. This is carbamic acid, and what you notice is the carbonyl carbon here, and then with the amine attached to it. And so if I start following and looking at all of these carbamates, right? So they have similar structure. If you'll notice, here's the carbonyl carbon, there's the nitrogen, right? If I move over here, there's the carbonyl, and there's the amine. And so following through, all of those have that same structural moiety. Yeah.
Is even carbamic acid and acetylcholinesterase inhibitor?
The way I understand it, yes.
So just even that small, like.
Yeah.
Wow, that's great.
So anyway, they're more water soluble. These molecules are generally more water soluble than the parathion, malathion, and so on.
So carbamic acid is, that's like it's almost an amino acid. It looks like.
Yeah.
So it seems like our body might make some of that, you know.
Yeah, I guess I see what you're
They're
saying
too
there.
far away from being poison.
Right. So, but you realize that we have metabolic function which has small molecules in it, and
right.
and so then you take those molecules with the appropriate moiety, they become dangerous, right?
Yeah.
Where other molecules aren't. And so yeah, that's the way I would respond to that, is that, I mean, chemistry is complex, and if it fits in an active site, small molecules, I mean, like aspirin, they have a ability to interfere in the COX-2 enzyme system, and so on, and they're not very big, and bring down fever and those types of things. But there's other molecules that would fit in there that would be toxic to that system, or would stick irreversibly, and so on, right? And so it's all structurally related. You know, we talk about over and over again, structure determines function. And so that's kind of what I was pointing out, is that those molecules all have some similarities, right? But there's parts of them that have been changed to make them less toxic for mammals and those types of things, where they're more toxic to the insect acetylcholinesterase than they are to ours. Does that make sense? You guys with me?
Yeah.
And so we see that those changes occurring, and as we understand structure better, now Dr. Easton will probably be able to have a better conversation with you about the synthesis of the structure and those types of things, and why those changes might have an effect with his knowledge of organic and biochemistry, right? So he could have a better conversation there. Other molecules, herbicides, paraquat, atrazine, 2,4-D, and allichlor, excuse me, are all molecules also that can cause issues, but have been used extensively in history. And if you look at the paraquat and diquat, and you see amine or nitrogen-containing rings, and these work as herbicides and interfere with plant function. So very popular back in the 60s and 70s. okay, and have since gone away. There's some maladies associated with those molecules, and the size of them probably interfere with DNA in that mechanism. I don't know that for sure. I'd have to look at the toxicity of them and where it comes from, but just looking at structure, that's what I would assume. And this is atrazine and metribuzine, and so these are for corn belt, no-till agriculture molecules. They're fairly stable, and they are water soluble enough they end up in drinking water, and so we're tracked there to those types of to aquifers and those types of mechanisms to give exposure to people and livestock and those types of things. And so those molecules were used in the Corn Belt. This one actually was heavily used in Vietnam also. This is 2,4-D, and this is a very popular broadleaf, and you can toxin, and you can see where it's used in the U.S. and those agricultural regions quite heavy. I sprayed a lot of 2,4-D. This was our favorite thistle spray, right? And to kill those types of weeds and so on. But anyway, 2,4-D and then the 2,4,5-T. And so both of these molecules are very effective for broadleafs. Very, very effective. I always tell a story about that molecule because I was spraying barn lots, cattle working pens, and we had, if you've been on the farm, you know that those weeds just show up everywhere. And so I was just trying to make everything look neat and tidy, and I had the pump started on the sprayer and was lifting the hose off the rack, and literally it blew off the top of the pump. It was already under pressure, and I literally took a full shower of 2,4-D there in the barn lot, and realizing that there was some risk with that, the hose was literally right there where I was standing, and so I turned the hose on and killed the pump, turned the hose on and just put it over my head, and then I started taking my clothes off and had stripped down to my underwear, and one of our gentlemen who'd worked for us for years, his name was John, he comes driving in the drive and drives up to the barn, and he says, "What in the world are you doing?" And I told him what had happened. And he says, "God, that must be cold." Right? So anyway, I went home and stood in the shower for another 30 minutes to an hour, and so far, I mean, I don't know of any effect from it. I mean, you guys know me though, but maybe I am a little bit crazy. So at least you're getting to know me. But anyway, that's a molecule that was fairly non-toxic to people, right? My exposure should have been high enough that if there was a toxicity to people. But could that have happened with a different type of chemical? Yeah, because we use that same pump, that same hand sprayer, and those types of things to spray a lot of different things. And so that risk is always there. So if we can make those molecules safer, right, and make regulations such that we remove the toxic ones, then we benefit people. Does that make sense? And I'm going to say on the farm we didn't wear the moon suits as we called them, right? Because farmers don't do that. Is that a poor choice? Well, with some of those chemicals, it probably is. The exposure is high enough that you should wear those types of things, that you should have a spray rig that has a filter cap that removes those molecules before they ever get to you, right? And so if we aren't smart enough to protect ourselves, then a little bit of regulation helps to protect us from ourselves. And so I'll just throw that out there in that fashion to think about it. This is a chemical that kills germinating seeds, right? So you can spray it to keep weed seedlings from ever forming. Alacor is the name of that molecule. And so effective in getting rid of broadleaves and grass seeds. Now these are some molecules, right, that we want to talk about a little bit. and these were never manufactured. Okay, this is dibenzodioxin, and this is the one that's quite notorious. The toxicity of it for some mammals is extraordinarily high. It's one of the more toxic chemicals that is known for some species. Now, it actually shows to be less toxic in other species, but for guinea pigs, 0.6 micrograms, right? 0.6 micrograms per kilogram of guinea pig. So 1 kilogram guinea pig, if you gave that guinea pig 0.6 micrograms, right? You had 10 of those, half of them would die at that dose. Now, micrograms not very much material. You guys realize that, right? So now, for humans, we obviously don't know the toxicity because it wouldn't be appropriate to study that on humans, right? But there are cases of exposure, and I want to talk about one of those. It's actually one of Missouri's historical legacies, right? If I mentioned the town before, I asked you guys if you know where Times Beach, Missouri was. Do you remember what? And you said no. And I said, have you ever crossed the Merrimack River on I-44 going to St. Louis? You guys ever crossed the Merrimack River going to, well, right there was a town or community called Times Beach. And the molecule is found in transformer oil, okay? So they used to use polychlorinated biphenyls in the transformers as a cooling oil, and when those oxidize, then one of the products that can be made are the dioxins, okay? And with chlorine present, then you could end up with this 2378 version of it. Now we could actually put these chlorines in different positions, or even more chlorines or less chlorines, and so we can substitute those out, but it's the right size to intercalate DNA. Right, and so that's what they believe causes the toxicity, and so then that molecule locks that up, and your cell can't reproduce the things that it needs to make, and so you pass right from that process. So anyway, and Times Beach is important because they took the PCB oil and sprayed it on the roads to keep dust down. Killed two birds, right? Got rid of the oil for the contractor that had that to get rid of, and it kept dust down. So what a great thing to do. You guys get that, right? So dirt roads, so they oiled them and no problem, right? Well, except that this molecule is highly toxic and it was in the dust. People were exposed to it, and so there were health issues associated with that, right? Higher levels of cancer, those types of things. So they took the top 6 inches of soil off of Times Beach, the entire community, and they ran it through an incinerator called the Blue Goose. And I remember when all of this was happening as a teen growing up, and it was a really big story, right? I mean, a legacy. It was one of the largest Superfund sites in history, and it's now a park today. So Times Beach is no longer there. It is now a park. The people bought that and paid to clean it up. Yeah. So, easily bioconcentrated. So, there's a town in Italy. I may dig out a video on that. I'm not going to today with all our fun looking for a cable. I feel like I got a little bit behind. And so we'll move on with part 2 of the lecture. And we'll start talking about some other things in water chemistry. And I put this slide in right here just as a reminder that we have nutrients that cycle through the environment and play a role in what goes on. We have decomposers and so on. But we can actually put in too much of these and cause pollution, right? And then if we have a biological oxygen demand, high carbonaceous material in the water, then you add nutrients, then we can really do some crazy stuff, right? In terms of a water system. So, well, in fact, we talk about things that consume oxygen, right? And so, number one thing I can think of is respiration, right? So that consumes oxygen when that's happening. So if I end up with a whole bunch of living stuff in water, and there's a whole bunch of food present, and there's the primary nutrients present, what happens? Things grow rapidly, don't they? And so then what happens to the oxygen? It goes down. So you guys are familiar with this. And so, well, when I also have nitrogenous waste and I have ammonia, then it takes the ammonia and turns it to the nitrate ion. And so that can use oxygen. When I have iron, we have O2, which that reminds me, we've been grading homework. We talked about you asked Garrett about the groundwater coming up, and we talked about iron, and if there's iron present, then oxygen would change the pH by producing acidity. But the other thing I didn't think about at the time when you asked me that question was there's CO2 present, and CO2, or even gases like hydrogen sulfide, which you can find in water. It's actually especially notable in, I know in Arkansas, for example, I have smelled hydrogen sulfide in groundwater coming out of a well down there at a cabin I stayed at. And so that molecule could be present, and both of those can leave the water when they come out of the ground. Does that make sense? Which then would change the alkalinity, and it's something I didn't think about when I was answering that question for you.
Okay.
Does that make sense?
It does, yeah.
Yeah, so I thought I'd mention that out loud to everybody, and that just came to mind when I was looking at this slide, and the iron and oxygen slide showed up, right?
Yeah.
And
So the CO2 now has access to sort of enter into that carbonate cycle.
well, or leave. So
Or
if it's
leave, yeah.
if it's coming out of especially high carbon rock carbonates, and so the carbonates are coming out of the rock, then in that equilibrium, CO2 can go in and out, so CO2 can leave, and so that changes the carbonate concentration in the water, if that makes any sense.
It does, yeah.
Yeah, so I just saw this, and because I think this is the reaction you were referring to,
I think so.
and it makes H plus in the process, so if oxygen's available, then it can use that to change the alkalinity. So anyway, sulfite is another reaction that could change the oxygen in the water. And so the other thing that we have to talk about is, can temperature be a pollutant?
Well, it can be a factor that contributes to.
It can be a factor that contributes to pollutant, or can it change the amount of dissolved oxygen in water? Yeah, and so if I have warming water, and then it can cause that to happen, I can lose gas, oxygen can leave, and things like turbidity in the water, right? So if I have suspended solids and those types of things, which will absorb more energy, make the water darker, and I can absorb more heat. And in fact, a lot of times in the spring, I always relate things to fishing. I can't help it, it's who I am, right? But sometimes you look for the cloudier water, like if there's a branch where floodings occurred, because that water will be 3 to 4 degrees warmer, and those fish will be closer to the bank. They move onto the bank, you throw a big spinnerbait or something that vibrates a lot, and you can catch more fish because of the warm water, right? So there's practical applications of that knowledge, yeah.
Is there a reason, like, with snow and, like, ice, that it turns, like, water will turn, I think, vividly more blue than, like going across the James River into Springfield when you're looking over the overpass
Right.
there? I've noticed that, like, when we get snow, it just turns, like, a brighter blue than it would any other time. I didn't know if there was a reason for that.
I don't know that water does have it is blue, right? In a sense, that's what you see. The other colors are being absorbed when you put materials in them, such as dirt and those types of things, silt. Then that silt can change the appearance of the water. maybe it's the blue water on top is that clean water that's landing there and staying to give that surface more of a blue appearance to you. I'm not sure, I don't know. I've never noticed that, but now I'm gonna watch and see if I see the same thing.
Notice it with like snow, and then like if you're looking at a spring, that water appears more blue if it's a deep spring, you know, than what you would see out of a deep running creek.
Depending on how clean the creek is, right? Now, if the water's really clean, then it has more of a deep appearance, blue appearance. I mean, you go to the tropics in the ocean, and I mean, it's very blue there, right? Especially like scuba diving, the red light gets filtered out by the time you get to depth. If you get to 75 to 100 feet deep, then everything is shades of blue, gray, and it's really kind of cool. It's one I'm a certified diver and have had the opportunity to dive in the tropics a few times. And the water clarity, for example, around the Cayman Islands the visibility is 100 foot. You can see the boat. You can look up and see it on the top of the water, literally. And I remember seeing an angelfish come swimming up. We were in a coral pinnacle, and an angelfish comes swimming up, and it was just a silhouette because there was no color. Does that make sense? But it was literally shades of blue or gray. For some reason, that image, I can always see that in my brain. It just floats to the top. It was just so amazing. But that's related to that color. I don't know. But yeah, water, it filters out light, but unless it has something else in it, and then it can change the color of the light, right? Because of that other. How do we put oxygen in water?
Aeration.
Aeration, that's a really good one. That's what they do in fish tanks, isn't it? They even do that at the dam at Table Rock. We've talked about that. So they aerate that water. How else do we put oxygen in there? Photosynthesis? Yeah, but now remember, those cells still are respiring, and so there's a trade there. But anyway, so when we start doing our measurements out in nature and we start doing our water sampling over here in the creeks and stuff like that, that's flowing water exposed to the atmosphere, and we'll find that it has 9 milligrams per liter or higher in that water. All right, so I want to talk about something called BOD, okay? Biological oxygen demand. There's actually a test to measure that. And the test is an interesting test. You sample water, and you measure the oxygen level in it when you collect it, right? And then you store that water based on a set of criteria for 5 days, and then you measure the oxygen content again. And so if nothing is using that oxygen, then the oxygen content should be very similar to what you started with, right? But if there is a biological oxygen demand under those conditions, then the oxygen will be used. Okay, you guys with me? And so this is a very significant test. It's used by, well, if we went to the water treatment plant, wastewater treatment plant at Springfield, then they would talk about that and how they use that to track what their oxygen demand is in their water, and then to make sure the water that they have clean doesn't have a biological oxygen demand, so that when they put it back into the environment to become what we would call Wilson's Creek, then we won't have a septic zone beyond that. I'll have to explain that in a second. Now, they actually, for operating, you can actually run a total organic carbon test to approximate that, which can be done very quickly, right? If you're trying to adjust your system based on a 5-day test, you're 5 days behind. Does that make sense? And so you have to have ways to monitor it real time, and then you use the BOD test to confirm, right? What you thought was correct in your changes in operation. Now, when I'm talking about biological oxygen demand, I want you to think about a point source. So I've got a slide here, and we're showing a point of waste discharge. And so we see it plume out into the stream. We're at the confluence. You notice that this part of the stream is still clean, that there has to be mixing that occurs to get that going. The slower the body water is moving and those types of things, the slower that mixing process occurs. But anyway, now what we want to look at is this from this perspective down here. And so here we're in a clean zone, right? And so the species that are up here are clean. And if this is a warm water stream, then it'd be warm water organization. If it's cold water, then that's what would be there. But it's a clean stream. You guys with me? Okay? And the dissolved oxygen should be normal for that type of stream because there's not a large biological oxygen demand. Okay? But when we put in that carbonaceous material from that point source, then where that happens, then what happens is there's bacteria that start eating that. You guys with me? And so when I have a whole bunch of bacteria there, I have a whole bunch of respiration occurring. Right? And so the dissolved oxygen plummets. And initially, the carp and the gar and those types of fish can live, the rough fish as we call them. The game fish can't survive here. And so then when we get to the septic zone, right? So the septic zone, then we have only fungi, bacteria, those types of things which are finishing up the cleanup process. And in fact, we're in anaerobic conditions, right? You guys with me? And then as those get cleaned up, what we see is that the trash fish can come back, right? And our oxygen starting to raise. Our biological oxygen demand is going down, right? Because we cleaned that up and then we make it on downstream to a clean zone and we can have our game fish again. All right. This is classic. This has been seen over and over and over again. Now, how do we prevent this in the real world? Do we discharge our sewage directly into the stream and then dilution is the solution to pollution? Or is there a better way? Have we changed what we do? This is what we used to do. What do we do now? We build a wastewater plant, don't we? And so we build a plant which we run this discharge through to remove all of the biological oxygen demand. That's the biggest job in a wastewater treatment plant. You might have thought it was something else, but that's really what it is, to get rid of as much of that as possible. Okay? And so then we can dump water in that is normal. So when pollutant discharge exceeds the flushing rate, water can become anaerobic. We can't support higher life forms. And so then beyond that, algae starts to grow, and bacterial degradation of the dead algae can consume O2. And so those nutrients are all there. So anyway, so that's kind of the scenario with a pollutant. Now there's other things that can happen, right? Dissolved oxygen and temperature caused a fish kill in Lake Washington that you can study. And so this is depth in meters, and this is temperature on this axis, and the blue line is the changes in dissolved oxygen related to that. And so what you see is that the higher temperatures, right, cause a diminishing of the dissolved oxygen. Oh, shoot, I got the wrong line. This is the dissolved oxygen here. So anyway, so what we see is that it comes down and comes back up, even related to temperature events. If you'll notice below the thermocline, and the oxygen there is very constant in both systems. But anyway, so we can see things like this that really affect temperature. We can also see where not only is oxygen related to fish kills, okay, but certain type of cyanobacteria or green algae are toxic. And so this is up in Washington State, and there's Lake Washington, and this lake, So Sanish Lake has had a number of algal blooms that produce cyanotoxin, and those toxins then kill fish, right? And so there's a whole tracking or monitoring of Samish and other lakes in the area related to this. And so it's kind of an interesting story if you get to reading about it. And so what they do, they monitor that, they know when it's coming, right? And try to affect that in a positive relationship. Now, these lakes are quite beautiful and used by people all the time. And so in terms of the management goal, one thing would be to keep the total phosphorus down, right? Because what happens when we produce lots of phosphorus and put it into the water? Causes those algal blooms to increase, and so they track that. And then they start tracking chlorophyll also, right? Because then that's related to the growth of the bacteria. And then they also track water clarity. Does that play a role in this? Yeah, so the clearer the water is, the deeper the light reaches. The deeper the light reaches, the more photosynthetic activity we can have and more growth, as long as there's nutrients present. And so all of those play a role in this, and you can see they've been tracking it for a number of years related to some of those, and there's a full management plan that you can read. I'm not going to take us there. I don't know that it's worthwhile, but if you're interested in this type of things, you're a wildlife conservation management major, then there's some really cool case studies related to these stories. And so anyway, just to kind of throw that out there, is it something that you might want to investigate a little farther. So anyway, so we kind of started moving towards this, sewage systems and those types of things, and the next chapter actually will go through how plants build, how it works to remove these types of things, and we'll talk about the Branson wastewater treatment plant and Springfield wastewater treatment plants. They have worked together and had a pilot study for removal of things like phosphorus and nitrogen from these waste streams in terms of that. But anyway, these are some of those constituents that we want to talk about. We've talked about the phosphates and the nitrogen containing things, but mostly we're removing the oxygen demand stuff, okay? That's the primary role of most wastewater treatment plants. If there's refractory organics, then we try to get those out too. You know, the dumping large amounts of oil down your drains well, they actually have to remove that at the treatment plant, right, and put that over into a digester. And some of those are actually toxic to aquatic life, and so we don't want those out. We try to get rid of viruses and bacteria which are harmful to humans. Household detergents, right? Grease and oil, salts, heavy metals, chelating agents. So if we can get rid of those, we try to get rid of those. Now, sometimes that's easier to do than what you think, okay? But we work towards that, trying to get it better. I always laugh, some of the solids that we've talked about in the number of years that I've toured that arrive at the wastewater treatment plant, you would be amazed at, right? But even in some of those larger drains, lawn chairs, how do you get a lawn chair in a sewage system? I want to know that. How would you park that there? But anyway, but all types of other things and in that relationship. All right, what can a sewage plant remove? Oxygen demanding substances and phosphates and some metals are removed through aggregation and colloids, and so that's really what's cleaned up. Nitrates are also in some of the modern systems, and with the right bacteria. Poorly removed refractory organics, poorly degraded organics that contain halogens are almost impossible to get out. That's the same thing with organics that we talked about in pesticides and herbicides that had chlorinated functional groups on them, right? And so those are very stable in the environment. And then viruses and pathogens are hard to remove, so what do we do instead? What do we do? How do we get rid of pathogens in wastewater?
Boil it?
You could boil it. that would work, but the energy expense to boil it, to get it to high temperature would be very, 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 very expensive, right? So instead we put chlorine in it, don't we? Or we put in ozone, right? So we mix that as the last step. I like ozone as a choice because it's used up fairly quickly and it doesn't chlorinate the remaining organics to make them cancer causing. Okay, so it's just something to think about. And radionuclides are very hard to get out. So anyway, I think I'm about done, but I'm going to do one more slide here before we go. I want to show this. This is the ocean here. If we take water, we put a discharge pipe out, then that discharge pipe comes out, and we can actually do that. Well, this was the way we used to treat sewage. You guys remember Dory, right? Yeah. Somebody swim up one of those to get to the dentist's office? Yeah, I'm done. I just thought it in class with that. So you can put a smile on everybody's face. Right?